light-dependent reactions occur in the thylakoid membranes, as that is where photosynthetic pigments which absorb light are located. We can summarize the stages as having three important functions: breaking up water into hydrogen ions, electrons, and oxygen in photolysis, the formation of reduced NADP, and the production of ATP from ADP and phosphate in a process known as photophosphorylation. Within the thylakoid membrane, there are two different photosystems. Photosystem 1 has a reaction center with a chlorophyll A molecule with the maximum light absorption at 700 nanometers. We call the reaction center P700. Photosystem 2 has a reaction center with a chlorophyll A molecule which has a maximum light absorption at 680 nanometers. We call this reaction center P680. In step 1, photosystems contain photosynthetic pigments that trap sunlight. Each photosystem will have many different chlorophyll molecules and accessory pigments. When light hits a photosystem, the energy is funneled to a single chlorophyll molecule known as the reaction center. Each pigment of photosystems are able to absorb light energy of different wavelengths, making each center more efficient. Photosynthesis is initiated by the photoactivation of chlorophyll A. The energy is absorbed by chlorophyll and causes its electrons to be released as they go from its ground state to an excited state or higher energy state. This causes the chlorophyll to become oxidized. The space left by these electrons leaving is filled by electrons released during photolysis. During this process, water splits due to light. It produces protons, electrons, and oxygen, which is summarized over here. In step 2, if we start from photosystem 2, the excited electrons are picked up by a carrier molecule called the electron acceptor and transferred directly along the electron transport chain, which is composed of electron acceptors. As the electrons move along the electron transport chain, the electrons lose energy, which is coupled to reactions that synthesize ATP from ADP and phosphate. This phosphorylation is driven by light, so it's called photophosphorylation. The electrons fall back to their ground state and fill the electrons lost from photosystem 1. From photosystem 1, the electrons are excited again and then move to another electron acceptor terminating in a coenzyme called nicotinamide adenine dinucleotide phosphate, or NADP, which with the hydrogen from photolysis reduces NADP to NADPH, or reduced NADP. As the electrons move through the electron transport chain, proton pumps use the energy to move protons from the stroma into the thylakoid space. As the protons accumulate in the thylakoid space, they flow back across the membrane down the electrochemical gradient through ATP synthase, which are the enzymes that form ATP. When photophosphorylation involves both photosystems 1 and 2, we call this non-cyclic photophosphorylation. It produces reduced NADP and ATP. There's another type of photophosphorylation called cyclic photophosphorylation, which only involves photosystem 1. So instead of electrons terminating in NADP, they loop around back to their point of origin. This type of photophosphorylation produces ATP but not reduced NADP. Hi, my name is Mr. Science, aka Salim. If you're new to the channel, please remember to like and subscribe, and for more teaching resources, you can visit my website at www.mrscience.co.uk.